Hello everyone. So a small topic today, immunoglobulins, antibodies. Look, immunoglobulins are plasma proteins which are belonging to the globulin group of proteins. Immunoglobulins have two heavy chains and two light chains. Each heavy chain is attached with each other with two sulfhydryl groups, while the light chain is attached to the heavy chain with a single sulfhydryl group. There are two portions of an antibody or components. One is the Fab portion, which is the upper Y-ended portion, and it has the antigen binding site on each aspect. While the lower side, which is the Fc portion, is actually the stalk-like part of the Y-shaped structure. As these antibodies are plasma proteins, the amino terminal is on the upper end, which is carrying a positive charge, while the Fc portion is the carboxyl terminal, which is carrying the negative charge. Now coming to the types, there are 5 types: G, A, M, D, and E. Remember, the light chains can be kappa or lambda in all of the antibodies, while the heavy chains in G are gamma, in A are alpha, in mu in IgM. IgD has delta heavy chain, while the E one has epsilon. The sequence which I told is the sequence of prevalence. IgG is the most common one, 75% of all antibodies. IgA is the second most common, 15%. IgM is the third one, approximately 9%. IgD is the fourth one, which is less than 1, roughly 0.2 in some books. And IgE is the least one, which is less than 0.01, like 0.004 it is mentioned in some books. Now, how do they act? What's their function? IgG, the most common one, it is always a monomer, first of all. Complement fixation, Fc portion of IgG, opsonization. Then it is also the antibody which is crossing the placenta from the mother to the fetus. And it has 4 types, IgG 1, 2, 3, and 4. IG2 is very important in encapsulated bacteria, fighting against encapsulated bacteria. Then we come to IgA. IgA can be a monomer, a dimer, or a trimer. IgG had a molecular weight of 150,000, while IgA has a molecular weight of 160,000. So it is majorly the antibody which is present under the mucosal surfaces, under the skin, present in our secretions, like it is also secreted in the breast milk, and it is delivered to the fetus in the early time of the life. And many pathogens which invade the mucosal surfaces and the skin, they produce IgA protease to fight against this antibody. Then IgM, the largest of all, it's a pentamer. The molecular weight is 150,000 yet again. And what are the functions of IgM? First of all, it is the only antibody which can be produced by the fetus in certain infections. Complement fixation, and as it is a pentamer, it can have up to 10 antigen binding sites. So it is the most efficient one of all. The fourth one, IgD, molecular weight 180,000. No specific function. It is present on the B cell surfaces, and it acts against under certain upper respiratory tract infections. Then the last, IgE, molecular weight 190,000 or 190,000, and it is present on the mast cells, the basophils. It is the major antibody fighting against allergic reactions, and yet again in the parasitic infections. I guess it comprises all of the antibodies. Thank you so much.